So today, the topic I'm going to speak about in the scientific seminar will be around the great unknown. And I use this title because in any environmental pollution situation, we're not exposed to just one compound, but realistically, a wide diversity of compounds, some of which we can identify, other which are very difficult to identify. So I'll share a lot with that, on that topic today. I want to start with some information about the new journal. Obviously, you heard my introduction. I'm here in Singapore and not so far away from my colleagues in India. Hopefully soon the pandemic lifts and we'll be able to visit each other once again. I'm joined by three initial associate editors that I appointed to the team. We have Qinghua Huang at Georgia Tech in the United States, Professor Jörg Dravis at the Technical University of Munich in Germany, and Professor Min Yang, who is at the Research Center for Eco-Environmental Sciences, which is part of the Chinese Academy of Sciences in China. But we're also really excited to have a very large international advisory board. We have people from all over the world, including we have a member in India, and we're looking forward to growing that group. So as the journal grows, you can expect that we'll increase the number of editorial advisory board members and, of course, more associate editors. So I very much encourage all of you to consider our new journal. I will personally try to help expedite your papers, and you'll see a very high citation rate as the journal comes alive. In fact, the journal began accepting submissions on June 2nd. We've already received well over 100 submissions, and the first ASAP articles online came live last night, so just last evening. And the first full issue will be published in early January. So we do cover a very broad amount of topics around the water theme, from treatment to natural hydrology to policy and regulation. So we do hope, again, to see a lot of contributions, particularly from India, with so many exciting developments and challenges in the water domain. So just a few examples. This is not comprehensive, but of course, water quality, chemistry, treatment, water protection and reuse, monitoring techniques, contamination remediation, risk assessment and regulation frameworks, as well as life cycle assessments. So hopefully, if you're interested, please go online and have a look at the new journal, and you can sign up for e-alerts that'll provide you information as the articles are published. And as you may know, we are part of a larger portfolio from American Chemical Society's Environmental Science and Technology, which now includes ES&T Letters, ES&T Engineering, and of course, my own journal, ES&T Water. So I'm going to move now into the scientific discussion. Obviously, here in Singapore and Southeast Asia, as well as in South Asia, we realize the critical importance of water for our economy, for our health, for the environment. And here in Singapore, our founding father, Lee Kuan Yew, actually made a very profound statement that every policy in the national agenda had to bend to the needs of water survival. And that challenge still exists, and I'll show a few examples today. So here we are. This is Southeast Asia, and we can see our neighbors here, our friends in India, just off to the left. In this map, you can't really even see Singapore because it's such a small country down at the very southern tip of Malaysia. But what may surprise you is that in some evaluations, Singapore has been considered as one of the world's most water-stressed countries. In fact, about 50% of our water supply is imported from Malaysia. And over the past decade, we've seen reoccurring droughts, irregular weather patterns, and a high degree of urbanization, particularly in Malaysia, which also stresses the natural environment. All in all, Southeast Asia, we have about 650 million people. And as you see here, the population has increased dramatically since the 1960s, with some of the countries in Southeast Asia increasing more than 4 times their population. Of course, the biggest populations being in the Philippines and Indonesia. For the small country of Singapore, we started out as about 582 square kilometers, but through land reclamation, we increased by about 23% to 722 square kilometers. And as I mentioned before, a lot of the water, about half the water, needs to be imported. And over the past few years, particularly in 2014, we exhibited historical levels of drought in the region. So in February of 2014, we had the lowest amount of rainfall since 1869. And this is despite a relatively high average of about 234 centimeters per year. For our neighbors in Malaysia, of course, they experienced the same drought. And the drought was so severe that it forced water rationing. Even parts of the nation's capital ran out of water and had to have water brought in by tanker truck. And certainly this isn't unique just to Asia. We've seen large-scale drought in Africa, which led to blackouts because of loss of hydroelectric, and same in Brazil. In the United States, of course, we've also seen very rapid changes in our weather patterns, from an example here of a reservoir in 2011, and that same reservoir shown in 2014. So these dramatic droughts have caused a lot of concern and a lot of new pressure on how do we become more sustainable in our water resources. So California has been leading the way, in part, at least in North America, in terms of developing plans for potable water reuse. That is, to take our wastewater and convert it into water that's safe and sustainable for drinking water consumption. And to that end, California has passed laws to mandate how they can regulate and protect public health as they're recycling their wastewater back into drinking water. But of course, this doesn't come without challenges. There's been a lot of concern among the public, and the area that I want to talk about today is chemical mixtures. So whether it's pharmaceuticals or endocrine disruptors, perfluorinated organics, the myriad of chemical products that could end up in water, if we're going to recycle or even just discharge it into the environment, how do we know that that water, when we use it for drinking water or protecting the environment, is actually safe? So we can roughly imagine that anything in commerce or even in nature has some propensity to enter the water supply. In the States, there's been a lot of concern around industrial chemicals. Here in Singapore, just about 1 month ago, there was a chemical spill in Malaysia for tetrahydrofuran that ended up even in Singapore's drinking water supply. And of course, there's been challenges even with the distribution of safe water, such as the lead issues in Michigan. So analytical technology today has become pretty incredible. Through high-resolution mass spectrometry in particular, both in liquid and gas phase, we can detect almost any compound you can imagine. But the real question is, are we looking for what matters? What's important to public health? So that begs the question, what is it we should monitor for? What is it we should test for? And how should we actually do that testing? So just think about any discharge, whether it's agricultural, industrial, or municipal water. We, again, have a very complicated mixture of chemicals. We're looking at an example in Southeast Asia. This is a satellite photo of Manila. And in the city of Manila, you can see the discharge of plumes that are going out into the sea, into Laguna de Bay. And it begs the question again, what is the composition of this mixture, and is it relevant to aquatic or human health? Photos from Lake Mead, Nevada. Of course, with higher urbanization and discharge of municipal waste into the reservoir under certain climatic conditions, we've seen epic algal blooms. And these blooms actually can include and did include toxic strains of algae such as Cylindrospermopsin. Another example from the United States is acid mine drainage, which has caused a great deal of environmental damage, and again can carry with it organic substances, heavy metals, and other contaminants as a complicated mixture of chemicals. So how many chemicals are there today? Well, that question has been raised many times. And the answer is going to be in millions of chemicals in commerce, and even more if we consider mixtures or byproducts, etc. And most of those chemicals have not been tested. In other words, they have not been evaluated for impacts to public health, and we certainly don't know the impact when they're occurring as a complicated mixture. And even if we were to employ, say, animal testing as we know it today, to try to evaluate the health risk, it could take centuries just to get through the current inventory of chemicals, let alone those that are developed every single day around the world. So taking me back to my beginning, I began working in this field back in the 1990s in a landmark study in Lake Mead, Nevada, where the US government had determined that fish impacted by wastewater effluents had shown estrogenicity, or that those fish exposed to various estrogens had impacts to their reproductive system. And this caused a lot of concern among the public, of course, that one of the largest drinking water reservoirs in the United States was being impacted to a point where the fish had deformities, especially to their reproductive system. So our early work was first disseminated in Environmental Science and Technology. Actually, on January 1st, 1998, there was an article that was speaking about our discovery. So we were able to positively identify molecules which were impacting the reproductive system of fish. So how did we do that? How are we able to figure out what the composition of this very complex mixture was that had already been demonstrated to impact the reproduction of fish in this particular reservoir? So our work, I'm very proud to say, was published in both Environmental Science and Technology around 1999 and in early 2001. And essentially what we were doing was developing methods where we could use instrumental techniques to quantify, identify, trace amounts of contaminants known to impact fish, but we were combining them with technology using human breast cells, which would respond to estrogen. So what we're showing in these figures are the chromatogram, in this case, LC fluorescence at the top, and at the bottom, we're showing the response of a human cell line to those extracts. The white bars that are vertical are showing fractions, so we collect a fraction of the chromatogram and expose it to human breast cancer cells and see how much induction of estrogenicity would occur. So on the right pane, you can see E2 and EE2, E2 being the natural endogenous estrogen that all mammals carry, including humans, of course, and EE2 is a thionylestradiol, which is a synthetic pharmaceutical used predominantly for oral birth control. So what we showed back here in the 1990s was that within this complicated mixture, we could best figure out the composition based on a combination of toxicology with human cell culture and analytical instrumentation which was predominantly mass spectrometry. So without going deep into this, I just thought I'd show one example of the cell line we used back in the 1990s. This is a human breast cancer cell, which has been genetically engineered with a luciferase gene. And that luciferase gene allows it to produce light when we add a chemical. So depending on the agonist upregulating the estrogen receptor gene, the natural receptor, we can quantify the amount of activity through a light production. And we still use that technology today. Here in my lab in Singapore, we have dozens of cell lines that we use, often with light as the reporter. So what about the impact to the people? Of course, this drew a lot of concern and have been asking, how can we advance this monitoring and do a better job of treatment and predicting chemicals entering the environment? And later, even considering that those concentrations of estrogens that we detected were able to cause extinction of some types of fish. So very profound impacts to the environment, and I think we can learn a lot from this example. So what's next? Where are we heading today? Well, for one, as I said before, we have millions of potential chemicals in the environment, whether it's pharmaceuticals, pesticides, household chemicals, inorganics like metals or oxyhalides, and of course, pathogens as well. So the chemical registry has over 100 million chemicals and substances in the registry. And just to give a grasp of this, in 2014 alone, more chemicals were added to the registry than in the combined years from 1965 to 1990. So how can we cope with that? How can we develop a comprehensive screening plan? Well, it has essentially two major components: targeted quantification, such as, say, WHO guidelines or lists of pesticides of interest. We can target them, we can use different types of analytical instruments, and we can very accurately quantify. But I think the more interesting, the more challenging world is this non-targeted screening. This is raising the question of how do we deal with the unknown, those things that we don't know what they are, but we can see perhaps that they're exhibiting toxicology to the environment. So for the state of California, I was part of an expert panel where we were tasked by the state to figure out what would a comprehensive testing strategy look like. So we suggested a three-tiered approach. Tier one are bulk surrogates. I'll show you an example of that in a minute. Things like fluorescence or UV, total organic carbon, conductivity. Often we can think of these as parameters that can be online in real time. The second tier, I already mentioned, we call these indicators. They are targeted analysis, and I'll talk about how we can pick a few indicators from the millions of potential chemicals. Then tier three, we do with less frequency, and that would be our non-targeted analyses and bioassays. So first, the evolution of analytical methods. When I was starting as a student back in the 1990s, we often use sample volumes of even 100 liters for one sample. That rapidly came down to about 1 liter. We began to do automated online extractions to reduce the sample volumes to about 1.5. And today we can directly inject water after filtration into our LC-QQQ and get parts per trillion quantification in just minutes. So the world has evolved very rapidly with the new instruments. So we'll quickly go through and show you just an example of what I mentioned before. So we can think about the different applications, again, pesticides or pharmaceuticals or natural hormones, but in chemistry, what really matters is the structure, whether it's a hydroxy aromatic or an alkyl aromatic or an aliphatic. And we begin to look at these different classes, and we can imagine that the fate and transport will roughly follow with those chemical structure. So we don't need to measure everything. We just need to measure indicators that are representative of the greater class. It's probably a little bit tricky for you to see this on your screen, but this was a paper we published in 2015, where we had been doing quarterly monitoring over a course of time at a wastewater treatment plant in the United States. And all we're doing here is we're grouping the chemicals that we detected by the concentration. So group 1 being very high, or usually parts per billion concentrations. And we're also looking at the frequency, which we have down at the bottom. So you can see chemicals like sucralose, which is an artificial chlorinated sweetener, are occurring at about 100% of the time in every single sample, and at concentrations upwards of 10 parts per billion, so relatively high concentrations, whereas the estrogens are occurring, you can see, at a very relatively low frequency and at very low concentrations. Now, that's not to say they're not important, because they certainly are, as we showed before in toxicology. One important thing to keep in mind, if we look at some of the most predominant chemicals that we can detect, in this case showing Arizona, then we compare that to Singapore, we can see that there's some overlap, such as asulfame, which is one of the artificial sweeteners, but there's also some big differences. For instance, in Singapore, we saw a propensity of iohexol, which is one of the X-ray contrast media used in medicine, whereas in the United States, we predominantly see iopamidol, which is another X-ray contrast media, but just used more frequently in North America as compared to iohexol, which is predominantly used in Asia. So how do we pick these indicators? Well, again, as I showed, we look for what is occurring at high concentrations. And we can also look at a particular treatment facility like wastewater, which is generally using biotransformation from the bacteria, but also absorption to the biosolids. So I have highlighted in red some chemicals that are very difficult to biotransform and do not bind well to the biosolids. So pharmaceuticals like carbamazepine or meprobamate, primidone, can very rapidly go through a wastewater treatment plant with relatively little attenuation. Same with the artificial sweetener sucralose. And we can begin to apply this for treatment processes beyond biological, such as ozonation, where we can look at chemicals that are very rapidly transformed by ozone, and then chemicals which are very weakly transformed by ozone, such as the iopamidol X-ray contrast media. The same is true for ultraviolet processes. Again, if you're interested, you can read the publications, but we can bin the different types of emerging contaminants into groups based on their kinetics, based on their quantum yield for photons, and we can begin to predict broader classes. We can also trace this through a surrogate measurement, such as the fluorescence I show on the right pane, and I'll talk more about that next. So again, let's talk about the surrogates, which are generally online in real time. And I'll show another example for ozonation. So ozone is a very important disinfectant. You can see it can convert healthy and happy, if you will, E. coli bacteria into a highly oxidized and dysfunctional, no longer living form. We also are using ozone to control fouling of membranes, but it's dangerous because we can actually destroy the membrane if we don't carefully control the process. I won't go into this in depth, but it's a very exciting area of study. And what we see is that different doses of ozone reduce membrane fouling. However, again, if we dose too much ozone, we will destroy the membrane. And that has happened even at full scale, where the application of ozone has converted bromide to hypobromite, hypobromous acid, and caused rapid deterioration of the membrane. So the question is, how do you control the system, especially when you're not adding a lot of the oxidant? And our answer, our recommendation, is to consider a surrogate like fluorescence. So we can see the excitation emission at the top in the normal wastewater effluent. And as we begin to apply ozone, we see a bleaching effect. So we can see a very quantifiable change in the fluorescence, which we can now use to control the ozone dose. So we've published this work a while back. We can not only look at the control mechanisms for ozone with fluorescence, which is TF on this slide. We can also use UV transmittance, which is very simple. And we can look at the predictive oxidation of a number of emerging contaminants from pharmaceuticals, personal care products, and others. So this is a really handy way to build a control system for ozone that's not relying on measuring the dissolved ozone, which is quite complicated and often unreliable at low dose. So what about the non-targeted? So sometimes as we're trying to fix something, we break something else. In this case, we were applying ozone in a water recycling plant to control biofouling on the membrane. And what we found was that as we applied ozone, we actually formed nitrosodimethylamine, or NDMA. And NDMA is a highly carcinogenic byproduct. The WHO has established a guideline of 100 parts per trillion. California has a guideline of 10, and the US EPA a staggering 0.7 nanograms per liter for the 1 in a million cancerous. So we were successful to prevent membrane fouling, but we created a new problem. So that's just one example. You can imagine as we add an oxidant to water, we can form thousands and thousands of different byproducts simultaneously. So what I'm showing here, this is a quadrupole time of flight instrument with a liquid chromatography interface. And I'm just showing the total ion chromatogram for the raw feed water, which is a effluent from a wastewater plant. Then showing 4 different doses of ozone from 1.5 to 5.6. So in the total ion chromatogram, it doesn't look very different. They look quite similar, actually. But if we begin to process this with statistical software, we actually see that these waters are very, very different. This is plotting the same data, but using principal component analysis. And these clusters are the different doses of ozone. So we can see that the structures are quite different and quite unique in each ozone dose. We can see it even better, perhaps, as we look at a heat map. So we can see that some things are formed, some things are removed, and the water quality is, in fact, changing very dramatically. So let's zoom in on just one example from that complicated mixture. If we look at the chemical benzotriazole, and triazoles are very commonly used industrial chemicals used as corrosion inhibitors, often in cleaning solutions or laundry detergent, and they're ubiquitous in wastewater effluent. So here we take a water with benzotriazole, one chemical, and we add ozone at time about 3.2 minutes. And we're doing this in real time. We had the mass spec connected to the reactor, so we can see the benzotriazole degrade, and we can rapidly see a byproduct forming. So using high resolution mass, we can demonstrate that the benzotriazole is actually converted into a dicarbaldehyde. But this only takes us so far. So we know it formed a new compound, but we don't know if that compound is relevant or not to human health, which is an important question that cannot be solved by a mass spectrometer. Here's another example, same thing, the idea carbamazepine, which is a ubiquitous pharmaceutical found in wastewater. And as we dose a small amount of ozone, we can begin to degrade or oxidize carbamazepine, and we form a very stable byproduct. Once again, through high resolution mass spectrometry, we can identify that byproduct as what we'll call BQM. But again, you can see the molecule remains relatively intact. So I only have a few minutes left. I wanted to cover this last piece on bioassays before I open for questions. So another piece of work we did about 4 years ago was published in Environmental Science and Technology on looking at non-targeted bioassays. So again, why would we do bioassays? So I've been working with this since the early 1990s, and the real advantage is it tells us about a larger class of toxicity. So while we can certainly do trace analysis of targeted compounds, the bioassay allows us to explore a deeper option of what is the relevance and is it toxic or not. I don't have time to go deep into this, but just to show you one example. So we began a program of monitoring wastewater treatment plants in Arizona in the United States. And I'm only showing you examples of glucocorticoid, which is GR, androgen receptor, the human male hormone receptor, and ER, which is estrogen, which I talked about before. So if you look at this bar graph, the red means logarithmic induction, very high levels of activity. And the black or dark blue means no activity or even a reduced activity. And so what we were surprised to see is a very high amount of glucocorticoid activity, which is one of the endocrine disruptors. And we noticed that every time we applied ultraviolet light, the activity went down. Estrogen activity, we saw a good bit at one of our very old wastewater treatment plants, but our newer treatment plants, really, we didn't see much activity. So let's take a deeper look at this GR, or glucocorticoid activity, because this was quite new to me. I had never seen it before, and I was quite surprised. So what we see is that glucocorticoids are among the most widely prescribed drugs in the United Kingdom, same with the United States. If you compare the amount prescribed compared to estrogen, it's about 10 times higher. So a lot of these compounds do enter into our wastewater treatment systems. And in the United States, these drugs have been approved for over-the-counter sale, meaning you can go into almost any grocery store in the United States and buy these types of products without a doctor's prescription. So what did we do? We knew that we had activity in the water, but we did not know what it was. So we would extract our samples, we'd analyze with the bioassays, we would fractionate by either size exclusion or by polarity, collect the fractions, analyze them again. And when the fraction had high activity, we would rely on high resolution mass spectrometry to try to identify the compounds. This is just showing you an example. Again, once we had the high resolution data, we could use that data to try to do identification. We were successful at determining some glucocorticoids that had never been reported in literature, so very much a surprise. And we were able to develop a very sensitive triple quadrupole method so we could more accurately identify and quantify at parts per trillion levels. And from that, we were able to do a scanning of different effluents in our region and to identify several novel glucocorticoids, including this particular one, triamcinolone acetonide. We'll call it TCA for short. And that accounted for about 50 to 75% of all the glucocorticoids detected. Again, I'm running out of time. I don't want to dive deep, but what was really important was to see if we could balance. And that means, could the chemicals we identify account for the toxicity we were seeing in the bioassay? So first, we had to test each of the glucocorticoids in our bioassay. We are quite surprised if you see this EC50, we can convert that into a relative effective potency. So how powerful is the effect against, we use dexamethasone as our positive control of 1. So you can see this TCA again, this one that I said counted for most of the glucocorticoids, was actually 2.3 times more potent than our positive control of dexamethasone, which we think is why nobody had previously been able to balance this, mass balance the activity. So in the end, we were very happy that we were able to see very good agreement between our glucocorticoids and the activity. You can see they trended together, the concentrations, and the in vitro activity being the solid line between. We also were able to see that we could remove the activity by ozone, although it took quite high ozone doses. And with ultraviolet light, we are also able to reduce, completely eliminate actually the bioactivity at relatively low doses of UV. So this is one of the rare chemical classes where ultraviolet turned out to be the most efficacious means of reduction, more than ozone, considering equal amounts of energy input. So that was kind of a rapid, if not a bit overview of some of the work we've done. I'll end on just a couple of photos for you to look at. Where are we heading? Today, we work a lot with zebrafish embryonics and genetics, doing things I didn't think were even possible when I was in graduate school. We are also, I think, very excited to be growing entire organs on a chip. So here at NTU, we've successfully grown human kidneys. So this is a human kidney organoid that functions just like a human kidney. And it's allowing us to really expand our relevance in terms of studying, in this case, disinfection byproducts known or attributed to kidney cancer, but without doing an animal test, in vitro with a human kidney, but without having to test an animal. So I think you're going to see increasing reliance on this idea of bioassay, and you're seeing more and more agreement between animal testing and the in vitro testing with just using the cells. So again, I went pretty quickly through a lot of material. So I hope I have some time here. We can take any questions about the journal or about the work, and certainly invite everyone to feel free to reach out to me as you wish. So thank you, and happy to take some questions.